Hi guys, today I'm doing a lab to test for the presence of specific hormones in panda urine. The purpose of this lab is to figure out whether the pandas are ovulating so that they could be mated with other pandas in order to save all the pandas. All right, let's get started. So first of all, in terms of materials, these are the samples. The P4, 2, 3, and 7 are the panda urine samples. And over here are the different controls, antibodies, and the substrate. I also have a little well strip, and this is wash buffer. I also will be using a different pipette for every sample, so here is my large box of pipettes. First thing I'm going to do is label my well strip. Now that that's done, I'm going to use a pipette to transfer 50 microliters of my positive control sample into the positive control wells. 50 microliters is really super small, so 25 microliters is the smallest notch on this pipette. So I'm going to do 2 of those super small notches. Now I'm going to repeat that same process again with my negative control and each of my panda urine samples. Now I wait 5 minutes for all of the samples to bind to the wells. These 5 minutes are perfect for a little yoga practice. Okay, back to urine. Next step is to dump all the samples out of the wells. Then I'll be using wash buffer to wash out the wells, and I'm going to repeat that a few times. Let's do it. All right, so I washed out my wells 2 times, and now I'm going to add in the primary antibody. Add 50 microliters into each well. Now we wait 5 more minutes. Perfect for a guided meditation. Now I'm going to dump out the primary antibody and then do a few more rinse steps. Now it's time to add the secondary antibody into the wells. The secondary antibody will bind to the primary antibody, which the primary antibody is bound to the hormone if the sample contains the hormone I'm looking for. All right, let's do it. Now, I wait 5 more minutes. This is the perfect amount of time for a tiny dance party. Okay, let's rinse them out. This time I'm doing 3 wash steps. Now I'm finally on the last step. I'm going to be adding the enzyme substrate into the wells. The substrate is for an enzyme that was attached to the secondary antibody. If the secondary antibody is still in the wells, then it will react with the substrate to turn the liquid blue. Let's see which pandas are ovulating. Now we can check our results. So here are my wells. You can see that positive controls are blue, which is a great thing. Panda 2 and Panda 3 stayed clear, and Panda 4 and Panda 7 turned blue. So that suggests that Panda 4 and Panda 7 are ovulating because they are expressing the hormone I was looking for, which means that they could help repopulate the panda population. Thanks for doing this lab with me, and save the pandas.